Hey everybody, how's it going? Thanks so much for tuning in once again. I'm Jason Wilson with Curious About Cannabis. Today I wanted to do a review video on a resource that you may be interested in checking out. One of my resolutions for this year is to try to make you a lot more content like this, where I'm reviewing resources that I've found interesting or valuable in my journey to learn about cannabis, and maybe it'll help you on your educational journey too. So today I wanted to explore the medicinal genomics tool called Kannapedia. It is a genetic reference library in some ways similar to another tool called the Phylos Galaxy, but quite a bit different, and we'll see that and talk about that shortly. But basically, this is a tool where a lot of genetic data about cannabis strains has been collected together to show how cannabis strains are alike or different, how common or uncommon they are. And it also pairs together chemical data too, which I really like. So anyway, let's jump right in here. So I'm at the Medicinal Genomics website, medicinalgenomics.com. And all you have to do is go to the Kannapedia link here. Click on that, and it'll go to Kannapedia.net. That's spelled with a K, Kannapedia.net, Kannapedia. And before we jump in here, we see that on the homepage, it's basically just a search bar. But let's go to the About, and I'll tell you a little bit about what this tool is and why Medicinal Genomics says they designed it. So this says, Medicinal Genomics created Kannapedia to publish genetic information obtained using the company's identification and registration service. And they provide the identity, heritage, and chemistry of cannabis and hemp plants that have been tested. For the first time, growers, consumers, patients, and clinicians will be able to identify the exact strain and key characteristics of the cannabis and hemp they're purchasing, enabling confidence in the quality and reliability of the product. So that sounds exciting. We'll see if it holds up, but seems like basically the idea is they've got this big genetic database, and they're allowing you to have a way to take a plant that you have, get its DNA tested, check it against this Kannapedia database, and see if it matches. Stop guessing, start knowing. Strain information is available for registered strains, showing the heritage, cannabinoid levels, consumer commentaries, and reviews. Well, I think this is what some of you will probably be the most interested in here. Protect your strain. So, Phylos, it's a cannabis genetics company based out of Oregon, where I'm located. When they started their company, this was the message they delivered as well, that they wanted to help people protect their strains by getting strain data into the public domain, so that if anyone ever claimed that they owned that strain in the future, they'd have some way to defend against that. Phylos has come under some controversy lately because of comments they made to investors about the future of the industry, and particularly the future of other growers, which were their customers. I'll just kind of leave that at that. You can look into that for yourself if you're interested in those politics. I'm not really so interested in the politics of all of that, and I don't really know what to believe and what not to believe, but I'll leave that to someone else. So they say each Kannapedia page here includes documentation that can be used to protect strains from corporations that may file genetic patents in the future and competitors who may claim to have your strain. We do this by publishing your strain's unique genetic ID to the Bitcoin blockchain, a disaster-proof web database to establish a time-stamped ownership record. Okay. I'm going to get some tea real quick. So that's cool. So they're using blockchain, which blockchain is basically a very secure ledger, basically. It has timestamps, and it shows that some things have changed, whether that be money in the case of things like Bitcoin and other cryptocurrencies. The blockchain shows at what time Bitcoins moved from one place to the other. So it creates a very defensible record of how money moved around. But you can use blockchain for all sorts of other things too. Any sort of data changes, you could use that for. So that's cool that they found that use for genetic data. They also have QR codes, it looks like, to link to your strain's Kannapedia page if you submit something to them to get tested. That's pretty common these days. All right, let's just go ahead and jump into this. So let's look at the phyllo tree first here. Oh boy. Okay, well, that's certainly not intimidating at all. Oh, okay. Wonder what font size that is. Well, this is tiny. Oh, look at that. Okay, so you hover over things, and it looks like it lights up the genetic path going as far back as it can to a common ancestor. That's cool. Just wish I could read anything that's on here. And it shows, looks like some genetic groupings and relatives, I guess. Ah, I see. Yeah. Okay. Well, let's read about this. So the cannabis phylogenetic tree, or evolutionary tree, shown below is a branching diagram showing the inferred evolutional relationships among various cannabis strains, or their phylogeny, based upon similarities and differences in their genetic characteristics. Okay. Here, the genetic characteristics are the identity and frequency of single nucleotide polymorphisms, also known as SNPs, in the captured regions of each cannabis strain genome. Genetic distance is a summary measure of the genetic divergence between cannabis strains, and the genetic distance of any two strains is approximately proportional to the total length of the radial parts in the path connecting the two leaves, which represent the two strains. All right. Well, that makes sense. That's basically a complicated way of saying what I was noticing here. Oh, this is interesting. So it covers 34 genes in the cannabinoid and terpene synthase pathway, which cannabinoid and terpene synthases are responsible for producing the enzymes that go on to produce cannabinoids and terpenes or other things. They also cover thousands of randomly distributed SNPs from the first version of their StrainSeq program, as well as looks like some other sources, as well as the Phyllis Galaxy. Okay, so this incorporates some data from the Phyllis Galaxy. That's interesting. As a result, a sample sequenced with this method can be cross-compared to all data that is public as of 2017. Well, that's really cool. I like that this is a compilation of data. It's getting a little old by now, 2017, but that's cool. So this is 25 times more sequenced than other tests on the market, and as a result, this is the most comprehensive sequencing tool for discerning clones from siblings and identifying uniqueness of a given strain. Okay, so it sounds like the main way they want people to use this is just to understand how unique your plant materials are that you have. And how related your clones are to one another, or other strains that you have to one another. Okay, here's something I was interested in. What do these colors mean? So hovering over a strain of interest will highlight the strain along with other strains whose genetic distances to the interested strain are color-coded as following. Red equal clones, orange equals siblings, yellow equals cousins, green is a distant relationship, and blue are things that breed with me for diversity. Okay, I guess those are things that are so distant. Yeah, that's an odd way to present that, but okay. So let's look at this again. Okay, so we see colors. We see mostly yellows, so cousins. But this is so hard to read. This is, I don't know. You guys tell me what you think, but to me, this is overwhelming to look at and to even consider using. I think it is intellectually interesting, but I am overwhelmed looking at this. I don't know if you are, but just like my eyes having to strain. I mean, I know I could zoom in and that'd probably be helpful, but I don't know. Let's go to strains here. Maybe we'll come back to this. Okay, so it looks like this is a listing of the strains that are in the database, which I assume and hope there are a lot. 28 pages. That doesn't seem like a ton. But maybe I'm wrong. I don't know. I need to look and see how many actual strains are in here. But I honestly expected more. But I don't know. I don't really know much about how long this has been going on or anything, so who knows. So XJ13, let's take a look at that one. So we've got a page here, kind of resembles like a certificate of analysis in a way, but with a lot of different data on it than you'd normally see. One thing I see that I like a lot is that they're assigning unique codes to every accession. That's super important because if you've listened to me talk on some of the podcast interviews I've done, I've mentioned that strain names are not very reliable in the marketplace. They can be, but they're unreliable enough that you really need to find ways to transcend them. And so regardless of the fact that they have a strain name here, you know, one person's XJ13 and another person's XJ13 may actually be not very related to one another, may not have similar chemical profiles. That happens for a number of reasons. Sometimes it's accidental mislabeling. Sometimes people fraudulently label clones and plants because the certain strain names sell better than others. And then also there's a thing that happens in cannabis breeding and other forms of agriculture too sometimes where growers will grow from seed, but then they'll label all of the plants grown from that seed as the same strain or cultivar as the plant that provided the seed. And now growing with seed, you're dealing with sexual reproduction. You're going to deal with a lot more, obviously a lot more genetic variability than if you were trying to do asexual propagation. So like cloning or tissue culture. So, yeah, strain names can be really unreliable. We need to find a way to transcend them. Obviously, this ID is not very sexy, but, you know, it is a unique identifier for these genetics, regardless of strain name or anything else. And that's really important so that we don't get caught up in a bad data set, basically, if we just rely on strain names. So happy to see that. We see that the rarity, it is common. So the number of strains, most distant, most similar. The strain rarity visualization shows how distant the strain is from other cultivars in the Kannapedia database. The y-axis, which is the up and down axis, represents genetic distance, getting farther as you go up. The width of the visualization at any point along the y-axis shows how many strains there are in the database at the genetic distance. So a common strain will have a more bottom-heavy shape. Uncommon cultivars will have visualization shifted towards the top. So this is very common. This is your super basic, super common, super average genetic. Interesting. High THC cultivar. Now this terpene data, first off, I'll say I love seeing chemical information partnered with the genetic information. That being said, recognize that this represents one sample that was sent. I assume it's one sample. I assume they didn't test multiple samples and average it together or something, but something I've mentioned before, but terpene data, it's so hard to interpret what to do with that data. I say take it with a grain of salt. Terpenes are so influenced by environment, and even after you harvest the plant, the way you dry it, the way you store it, if it's exposed to light, all sorts of things influence the terpenes that are in the product. So you could theoretically get a clone of this. You could tissue culture this plant, get it as genetically similar as possible, and you could still see different terpene results depending on how the material's handled. So this is cool. Just don't get too caught up in this data. And let's see, genetic information. Heterozygosity. So heterozygosity would be sort of like how variable the genetics are. It's sort of a measure of how inbred a line would be. So something that is less heterozygous would be more inbred and would be unlikely to produce a lot of genetic variation, whereas something that is more heterozygous has more diversity. And thus, if it's propagated, that diversity will come out. Let's make sure that I'm right about that. The bell curve in the heterozygosity visualization shows the distribution of heterozygosity levels for cannabis cultivars. The green line shows where this particular strain fits within the distribution. Heterozygosity is associated with heterosis, aka hybrid vigor, but also leads to the production of more variable offspring. When plants have two genetically different parents, heterozygosity levels will be higher than if it has been inbred or backcrossed repeatedly. Okay, so this one's a little more heterozygous than the average. So that's somewhat interesting. Maybe it's more interesting to you. It's cool. Let me get some more tea real quick. Okay. So nearest genetic relatives to XJ13. Of course, this is nearest genetic relatives in their database, but ooh, here's something very close. Wow, this Jack Herrera. I'm not sure that's the way you actually spell it. So we see clone distance. So that makes me wonder, does this mean that there was a clone made of these genetics and was called Jack Herrera rather than XJ13 or vice versa, since they're in that clone distance? I'm not positive that's what this means, but that's kind of what it seems like to me. And then you've got siblings over here. Nearest genetic relatives to the base tree. I honestly am not sure exactly what that means. If someone knows, explain it, because I don't see something that explains it here. Most genetically distant strains from XJ13. So what are the things that are most not like this? Apparently this Carmagnola, am I pronouncing that right? That's popping up several times. Apparently it's very, very different. There's the QR code, and there's the blockchain transaction ID. Cool. Now I saw a button up here I want to check out. View the strain on the phyllo tree. Let's check this out. It did not highlight the strain on the phyllo tree. And I do not want to hunt it down. Oh god. Every time I see this wheel, I'm just intimidated. All right, let's hunt it down. Let's see. So, where's the ID here? We'll copy this. And then let's go back to the tree, the intimidating phyllo tree. I see why Phylos did a galaxy. Copy and paste that. Oh man, we lost it. Okay. So I'm gonna have to get really close to the screen because my eyes suck. Let's see. Okay, I think I'm hovering over the right one now. So now we see all the immediate siblings there highlighted in yellow. And then we see it trace all the way back to the common ancestor. That's cool. We see the very distant relatives in blue on the almost opposite side of the wheel. Cool. All right. Just wish it were easier to use this. It's like an interesting tool, but I don't know. Let me know in the comments what you think about this. I just don't know. I don't know how I feel about this. I don't know if it's actually valuable or not. Like I said, it's interesting, but is it valuable? I'm just not sure. I want to do a search now. I'll show this. Let's look for moonshine haze. That's one of my favorites. Moonshine haze has a very, to me, or at least the instances of it I have come across have a very interesting terpene profile. The smell is unique to me, and I like it a lot. So that's why it stood out. Like I said, I'm not a breeder. I don't remember a lot of strain names because most of my work has been in looking at the chemistry of cannabis plants, and we don't really worry too much about strain names there, and they don't mean a whole lot. So I'm just really bad about remembering strain names. So just bear with me there. But moonshine haze is one I know, and it is uncommon. That's cool. Oh man, no terpene data. I'll have to find some terpene data on that and talk about it, because I don't remember what the common terpenes are in moonshine haze that give it its unique smell, but it's a little more heterozygous, more so than the other one that we were looking at. So it is not what some would refer to as a stabilized line necessarily. Somewhat stable, not as stable as it could be. Genetic relatives. Now, correct me if I'm wrong, Cannasu and Cannatonic, those are CBD strains, right? Let's see. Cannasu. Also uncommon. Oh man, no cannabinoid data. Pretty sure Cannasu's a CBD strain though. Let's look at most genetically distant strains. Blue Dream. Interesting. Carmagnula again. What is this Carmagnula? Let's check this out, because it keeps popping up as a distant relative for both things that we've looked at. It is uncommon. From Colorado State University. Interesting. No chemical data. Not as heterozygous as the moonshine haze. Looks like we've got some clones in the database and some siblings under the same name. Oh, what else is it similar to? Cannasu, Space Oddity. Interesting. I've just never come across the strain. And doesn't have a transaction ID. Weird. Interesting. So the quality of data, it looks like, in this database varies. Not everything has every piece of data that you might like it to have. But I don't know. What do you think about this tool, Kannapedia? I think it's interesting. It's got some potential. I think there's some aspects of it that are a little technical that some audiences might struggle with. You know, I have a biology background, so I'm not a genetics guy by any means. I really want to interview some geneticists. I've actually reached out to Medicinal Genomics, haven't gotten very far with them in trying to do an interview, but I really would like to interview some folks that understand genetics a lot better than I do. But I do have enough of a background in genetics that I can make sense of, you know, what's being presented here for the most part. But I think some people would really struggle with sort of the technical language and everything here. It's obvious that this was built by scientists and folks that are technical thinkers, and that's great, but sometimes you need a little help translating that sort of stuff down to a lay audience. And I know a lot of breeders do have a really good understanding of genetics on a technical level, but a lot of them also don't. So if they want breeders to use this as a tool, I think it could be simplified a little bit without sacrificing the integrity of, you know, what they're trying to do. Also, man, this phyllo tree, I think this is a noble effort. This is a cool thing, but I don't know. It just doesn't seem that functional to me. I don't know. Let me know in the comments what you think. So this is Kannapedia by Medicinal Genomics. If you like the video, make sure to hit like and don't forget to subscribe to the channel and hit the bell icon to get notifications. And if you have other tools that you found valuable that you'd like me to review, react to, send them my way. I'd love to check them out. So go check out Kannapedia and go play around with some strain data and let me know what you learn. And if you catch anything else in this tool that I didn't catch, once again, let me know in the comments. Thanks so much for watching and take it easy.